My name is Ariana and my lab partner is Austin, and we are presenting our Bacteriophage EnderDragon that we isolated from a Mycobacterium filarium culture. So a bacteriophage is a virus that infects and replicates within bacteria. So we used M. filarium bacteria as our host. And there's two cycles that bacteriophage can go through. So the lytic cycle, the virus replicates inside the bacteria using their mechanisms, and eventually they cause the cell to lyse or burst. In the lysogenic cycle, the virus incorporates itself into the DNA, and the cell replicates the bacterial DNA as well as the viral DNA, and it goes through cell divisions, and the cell can enter the lytic cycle at any time.
So for our bacteriophage, we isolated it from soil underneath a rock next to a tree in Glen Rose. And after plaque assays and serial dilutions, we were able to purify the sample and get a single phage. And from those, we were able to make webbed plates to create a high volume lysate with a concentration of about 1.5 times 10 to the 9th PFU per milliliter.
So after getting our high volume lysate, we moved on to DNA extraction. So after we extracted our DNA, we purified it, and then we put it through the nanodrop, and we got a concentration of 133 nanograms per microliter. This was considered low, but it was enough to pull through with the rest of the experiment. So we also did a qubit reading and got a reading of 16.6 nanograms per microliter. So we got an average, and we decided that our DNA concentration was 75 nanograms per microliter. So after we got our DNA, we added the restriction enzyme, and we followed through with the protocol for gel electrophoresis. And this is the results. So they all line up on this line.
After we ran the gel electrophoresis, we did a transmission electron microscopy and determined that our bacteriophage was Cephaloviridea morphology with a caspid diameter of about 69 nanometers and a tail length of about 158 nanometers.
So for our next steps, we are wanting to sequence EnderDragon, to annotate the genome, and to further analyze and characterize EnderDragon.